Okay. We're on specific immunity. It shouldn't take that long because, because we went over most of the chapter. You know what? There's something I forgot to talk about though, and I don't see it. I don't see it on here. Oh, it's coming up. That's why. All right. So, specific or acquired immunity. When we get our COVID vaccines, that's the part that's addressing, right? So there's two sides to it. There's the I'm going to go back a slide in a second. There's the this is what I was drawing on the board last class. There's a there's a B cell, an antibody mediated side, and then there's a cell mediated side. So I imagine it as like a two-pronged attack. So you're going to have T8 cells on one end and you're going to have B cells on the other end. And then you've got these T4 cells, which here they're calling them CD4 cells or they're calling them helper T cells. You can kind of think of them as T4 cells or helper T cells, right? It's whatever, it's fine. So they're kind of helping. They're Nothing's going to happen really without them. You notice that there's like a line saying help and it's going over here to the antibody side, and then there's a line here going to the T8 side or the cell mediated side, right? So it's like what's cell mediated versus antibody mediated? What's doing the attacking? What's attacking the pathogen? Are they cells or are they antibodies? Antibodies don't necessarily destroy cells, but they help. Without it, the T8 cells couldn't work. So we have three cells that are involved here: T8, T4, and B cells. Right? B cells is on the antibody side. T8 cells is on the cell-mediated side, and then T4 cells are helping with both processes. And it's kind of summed up here, right? The T8 cells are going to clone, and they're going to make a bunch of cytotoxic T cells. The B cells are going to, when they get provoked, they're going to become plasma cells. So B cells become plasma cells, and plasma cells secrete antibodies. That's what I have. That's what I have here, right? So B cells will clone into a lot of plasma cells, and those plasma cells secrete antibodies. And then we had the T4 cell in the middle. They're releasing a chemical that's going to assist in both the cell and cell mediated and antibody mediated. So before we get into that, antigens. Remember, antigen is like the piece of the pathogen that's going to that our immune system's going after. We don't go after a whole bacteria or a whole virus. We just go after pieces that are usually on like their protein coat, right? And so those pieces, or like the name tag of the bacteria, those are antigens, and they have to have two characteristics, right? So they have to be able to provoke our immune system. That's immunogenicity, right? Because sometimes, like I was telling you, like a virus will leave a cell and they'll wrap themselves in our membrane. Right? That wouldn't happen. That would be something with no immunogenicity. Our cells could attack it if we knew to attack it, but we don't know. So it lacks immunogenicity. So you have to have immunogenicity, and then you have to have reactivity. You have to be able to react with the antibody or the cell. Like once the cell or the antibody gets there, you have to have some kind of reactivity. And that doesn't always happen because, like, I was giving you another example, like when you change the epitope, when you change your antigen before the T cells or the B cells, like once the T cells get out there and you've changed your antigen, then you have to start over again. Right. So if something's a hapten, they have reactivity. It could react, but no immunogenicity. So it doesn't provoke the immune response. It would react, but it doesn't provoke it to begin with. So the epitope or the epitop, um, they're small. They're like the small pieces that provoke the immune response. That's one classic way to deliver vaccines. You just deliver those. You just inject somebody with those little fragments, and that's all you need. That's gonna provoke an immune response. I don't know why that photo's. Hope that photo's helpful. Don't know why it's there. All right, so we also were talking about this MHC, major histocompatibility complex, or major histocompatibility antigens, MHC, right? And they're in our plasma membranes. Some of them are on top, sitting on top, and some of them are built in to the membrane, right? So MHC1, they both do the same thing, right? It's just that, I don't know, I've seen test questions, not me, but I've seen test questions ask about like MHC1 versus MHC2. It's just that MHC1 are built into the plasma membranes. Of course, our red blood cells don't have this whole setup. They're using a different setup, the A and the B and the O. So they're not using this, but most of our plasma membranes have MHC1. MHC2 are on these antigen presenting cells that we're going to talk about, and they're not embedded into the plasma membrane. They're just sitting on top of the surface. So, really? So let me go to this slide first and then I'll back up. And again, this is from last class. This cell, this whole yellow blob is a cell. It's looking elongated, right? But it's a cell. It's a cell that it's called an antigen presenting cell. So it's a cell that presents the antigen, right? You have here the two main actors, T8 cells, and you have B cells. They have to be stimulated. They have to be shown that there's an infection. They're not going to just start cloning and go run at nothing when they don't know what it is specifically that's infected your body. They're not going to just run at a piece of pollen. You got to show them what specifically is infecting you. And so how do we do that? We have these antigen presenting cells. They're going to show you specifically what is infecting you. And so this whole yellow thing's a cell, and it's a phagocyte, right? So it eats other things. So we pretend that this antigen or this thing here, this spring thing with these shapes, that we'll pretend that that's a bacteria. So it's going to eat, step one, eat the bacteria. There it is. Step two, digest it, break it apart. Step three, take its pieces and use it in your MHC molecule. Remember, MHC molecule is like the name tag for your cells. They're very specific for your cells. So now you're making that with parts of another, like with parts of another, with parts of a bacteria or parts of a virus, right? So you're making the name tag that says, "Hi, this cell belongs to Klaus," and it's got parts of the bacteria built into it, right? So then when it makes its name tag, it's using those pieces from that bacteria. So that's all it is, right? And so this cell, after it's done, it's going to go walk up to where all the T8 cells are hanging out. And it's going to be like, hey, T cells, check out my name tag. Right? That's going to get them all riled up. So they must, for T cells or B cells, for T8 cells or for B cells to start getting revved up and get ready to clone, they have to see that there's a foreign antigen. Right? So here I'm saying T cells must have antigens packaged and presented to be recognized. That's the antigen presenting cell. They're going to take that bacteria or that virus and break it up and use it in their MHC molecule. And then they're going to show the T8 cells. B cells, they could do the same thing. They can use the antigen presenting cells. Like they'll see the antigen presenting cells and that'll trip it up. But also they might see like pieces of a bacteria in your lymphatic system or even in your blood, right? So they're if it's in your spleen or your lymph nodes or lymphatic nodules, that'll trigger the B cells there too. So the B cells have a couple different ways that they can get triggered. But in either case, the B cells or the T cells have to recognize that there's like a foreign antigen in the body. And so what do the T8 cells do? Now I'm like looking at this one, like, why do I have this whole thing? That looks a little complicated. Let's talk about what the T8 cells do. So when the T8 cells get activated, when they see that antigen presenting cell, they start cloning. Right? They start cloning and, well, actually, I shouldn't say that. They're getting ready to clone. But before they can start cloning, they have to get a stimulation from the T4 cells. And that stimulation is it's a chemical called interleukin-2. It's a type of chemicals that we call cytokines. Right? So interleukin-2 is a chemical that's released. If we go back here, it says like help. Does it say help or is that like an? Doesn't look like an E. Anyway, it's supposed to say help. So like this line is saying help is going from the helper T cell, and there's like this line going down to the T8 cell. That's interleukin-2. Same thing on this side. It's interleukin-2. Right. So the T8 cell is going to get stimulated twice. First stimulation, it sees the antigen presenting cell. That gets it all revved up. It's getting ready to clone. Then it gets the chemical interleukin-2 from the T4 cell, from the helper T cell. And then it could start cloning. So they'll start cloning and they'll become cytotoxic T cells. So now we got like millions and millions of cytotoxic T cells. And what they do is the same exact thing that natural killer cells do. They have perforin and they have granzymes. So they use those two weapons. So natural killer cells are similar to cytotoxic T cells, right? In that they use the same weapons. Where they differ is that cytotoxic T cells are for a specific immunity, right? They only know, cytotoxic T cells only know how to kill that one antigen. So if you get the flu, if you get tuberculosis, whatever reason I thought of that was, these won't work on it. These specific T cells are for that one infection, right? Whereas natural killer cells, they could go after like lots of different things, but they're not good at going after large numbers of them. Now, B cells, antibody mediated. So we have the other side, the B cells. The B cells are going to either see the antigen in the lymph or like the plasma or something, or they'll see the antigen presenting cell. either way, they also start getting revved up, but they also need interleukin 2. The same thing. They need that message from helper T cells. So once you have both stimulations, they're going to start um, becoming plasma cells, and plasma cells are going to secrete antibodies. Antibodies are not going to attack the cell directly. They're going to attack the cell indirectly. How? By these ways. I'm going to go right back to this, but let me jump to something else because when I see here viral attachment, it made me think of something that I forgot to tell you. Look, this antigen presenting cell ate this bacteria. So, and if you look down here, there's that word exogenous, right? Meaning that it's attacking from the outside, right? Bacteria, parasites, fungi, whatever it is, they attack from the outside. They attack from the outside of the cell. They try to break through the cell and they get into the cell. Viruses are different. We call them endogenous. Endo meaning they go inside first, then they attack. So they're a Trojan horse, right? So they go inside first, then they attack. So they get processed a little bit different. You don't have to eat. If this was a virus, you wouldn't have to eat it. It would just, so we would get rid of number 1 here. It would just go into you, and we could start with number 2. Right, so an antigen presenting cell will, the virus will infect it, then it will tear it apart and start using its pieces to make the MHC molecule. So viruses are a little bit different, right? Viruses are endogenous. You don't want them attaching to your cells, because when they attach to your cells, they're going to inject their DNA or their RNA. They're different. Viruses could either be DNA viruses or RNA viruses. But in either way, you don't want them injecting that into your cell, because that has instructions to make more of that virus. Right? So if you can block their attachment somehow, then they're not going to be able to spread. Viruses can't spread. Like, you can't take a bunch of viruses and put it on like agar or like feed it food. It's not gonna. It needs a living host. You have to inject it into. You have to grow it using live cells. So maybe you could use something simple like eggs, like chicken eggs, right? So it needs live cells to replicate. It can't replicate without a live cell. So neutralizing antigens, preventing viral attachment, that will stop viruses pretty quick, and blocks bacterial toxins. It's not the bacteria. When you get infected with bacteria, one of the reasons you feel sick is because your immune system is going, right? That makes you feel a little bit sick. But then the other type of sickness you feel is because of the toxins that they're producing. So bacteria, just like we do, bacteria, similar to us, they eat and they poo, right? So it's the poo, that's the toxins, right? Think about what causes us cavities. It's not the sugar, it's the bacteria that live in our mouth that you can't get rid of it, so don't even try. Bacteria live in our mouth. They eat sugar. They don't like eating proteins. They don't like eating fats. They like sugar. They eat sugar and then they poo out acid, like their whatever they excrete is like acidic, and that is what's breaking down our teeth, right? And so similar in our body, they excrete stuff when they eat. They're eating stuff in our body, and then they excrete stuff, and then that gets us sick. With pathogenic bacteria, I'm not talking about like the regular symbiotic bacteria that live in us. But anyway, if you could block that toxin, or if you can prevent viral attachment, that's going to do a lot. they immobilizing bacteria. So antibodies stop. Usually, bacteria don't move around with flagella, um, but they do use cilia a lot. And cilia are like hairs, like little hairs all around the bacteria. And they could use that to get around. And it makes it harder for us to take them out because they're constantly moving around, right? But if you could stop the cilia from working, then they can't move. Now you have a real advantage. Guys, stop being late. Throwing off the feng shui of the lecture. I'm using all my brain power to try to think. All right. Agglutinizing, bringing them together, clustering pathogens. That makes it a lot easier for not only phagocytes to get to them, but T cells, right? So it's like you're chopping off their legs, you're bringing them all together, and then look at this last one, enhancing phagocytosis. You send up a flag, right? So now you just look out in the horizon for a flag, and then at the base of that flag, there's going to be like a group of of pathogens that can't move. That's so much easier to kill them now, right? So when you look at what like an antibody, the molecular structure of an antibody, it's got like this stem region that looks like a flag. So when they grab onto these things, it's like a flag. So that's what they do. They neutralize antigens. They mobilize bacteria. They agglutinize or gather different pathogens, or here I put antigens, and then enhancing phagocytosis by marking the pathogens. So that's what antibodies do. That might be my last slide too. Some of these, I'm going to go back to here. Some of these killer T cells and some of these T cells and some of these B cells are going to remember the battle that you went through, right? Just like World War II vets, in case we forget what Nazis look like. And for all the bad things you can say about Nazis, they were pretty easy to spot, right? You're not like out in society saying, oh man, is that a Nazi? I don't know. Who's a Nazi? Well, they're white, and then they got like these red armbands with swastikas. So anyway, but the memory B cells and the memory T cells are like veterans, right? They remember the war and they remember what the enemy looks like. So it's not that you don't get infected again. Like if you get the flu and then you pass it to a family member and then that family member passes it back to you, you're not going to get sick. It's not because you don't get infected. That virus did go into your body again, and it tried to get things going again. It tried to start another war. But as soon as it got in your body, those memory B cells and memory T cells took it out. They're just like, hell no, I remember this. I remember this from like 2 weeks ago. Right. So those are, when we're talking about your immune system, I mean, that's what we're talking about. Right. And when you inject somebody with a traditional vaccine, not like the Pfizer and Moderna, but like the Johnson and Johnson, when more traditional vaccines, you stimulate the immune system, you get this whole thing going, right? They're building an army, they're preparing a battle. Turns out there's nobody to fight because they didn't actually inject you with a virus. They just injected you with pieces, like with antigens. They injected you with like the epitope, right? And so it wasn't actually the virus, but it doesn't matter. They consider that a war, and most of those killer T cells are going to die off, and the antibodies, some of them will stay around. A lot of the plasma cells are going to die off. The B cells that participated in that particular battle, they're going to die off, but some of them are going to stay around and remember. And that's the whole point of getting your immune system going, so that if you ever did get infected, there's not like this race. You don't have to start getting your immune system is going to take it out as soon as it like hits the body. All right. Yeah, that was the last slide, right? Yeah. So that's it. So, okay, so the main thing that you want from me. Test questions. Yes, be nice if you at least knew what the lymphatic system did. They and it's those three things, right? They drain excess interstitial fluid. I don't care about up to 3 liters more filtration than reabsorption. I mean, you should know that, right? You don't have to put that on a test. That's an example. It's me expanding on one of the functions, right? More fluid leaves the capillaries and goes back in. So your lymphatic system takes that extra fluid, transports lipids, and it's involved in immune responses. This whole thing that we've been talking about with the specific immunity, right? That's one of the things that sets off the immune system is your lymphatic system. Skip all this. Probably not going to ask this. This is a separate. I mean, you should know these to talk about other slides. Oh, you know what? I might ask you about significant pathogens. It'd be nice to know different types of things that can make you sick. Right? A pathogen is something that makes you sick. So what are different things that make you sick? This is not all inclusive, but these are the main things, right? Most of the time, it's going to be a virus or a bacteria. That'll be like, I'm just guessing here, like 90%, 95% of the time, right? And then if you add fungus to it, it's going to be like 98% of the time. Very rarely are you going to have, hardly ever would you see protozoa or parasites in the United States. If you did, it would be somebody that like traveled to a foreign country. You know, they go to somewhere in the developing world and like, oh, street food, that looks delicious. And they try all the street food and, you know, they're like, oh, raw, whatever. Oh, that sounds fun. All right. People that call food fun, right? They're going to be the ones with the parasites. That looks fun. Raw crabs, that looks fun. Why? We've got like the best crabs in like the world. And that sounds kind of weird. But you know what I'm saying? We have the best seafood in the world. You don't have to try raw crabs from somewhere. But anyway, those are the people that end up with parasites. All right. So significant pathogens, like what are pathogens that commonly infect people? No. Yes. So here we're talking about general defense. 2 types of general defense: first line, second line. So I'll ask you probably a question about, first line of defense, and this is a lot of things here, so you don't need all of these, but maybe if you knew half of them, if you could give me 5 examples, that would be nice. So, yeah. Second line of defense. That's going to be a little bit more, you're going to need a little bit more elaboration on this, right? So I don't want just the 4 types. It's not going to be that simple. You can't just list them. What do you mean by cells? Like, yeah, put cells, but what do you mean by cells? What kinds of cells? What do they do? Natural killer cells, phagocytes. What do those 2 cells do? So, you don't have to like do it exactly like it's on this slide. Just phagocytes eat pathogens. That's it. They eat stuff. Natural killer cells uses those perforin and granzymes. What does perforin do? What do granzymes do? Apoptosis is like cell suicide. It's like another way of saying cell suicide. And then the antimicrobial proteins. I want you to talk about those. And I want you to talk about fever, and I want you to talk about inflammation. I've got both of those on this slide. Right? Not just inflammation, but you kind of have to know what is inflammation, what's going on, why do things get inflamed? Because of increased permeability, of things. Increased permeability is increased filtration. And then dilating your blood vessels is going to move blood around or move blood to that area faster. And then the chemicals, because I think you're going to come across them again. Whenever you do that part on allergies, for example, you're going to, anyway. So, what about each of these four? So I think we're on 4 questions so far. What functions of the lymphatic system? Question 2, types of pathogens. Like there's 5 types: bacteria, viruses. That's 2. 3, first line of defense. Like list, and that's just listing. It's really nothing to talk about. List 5 different types of first line of defense. Discuss the second line of defense. So that's question 4. Discuss specific versus discuss specific immunity. That's a very general question, right? But so you'd have to explain, and that's a lot of these slides all. So you can actually, if you're going to discuss specific immunity, then you have to break it down. Okay, I'm going to have to talk about cell-mediated immunity, and I'm going to have to talk about antibody-mediated immunity. Or I might just ask you about like cell-mediated. Okay, you're probably confused. I'll ask no cell-mediated and no antibody-mediated. So on cell-mediated, what are the 2 stimuli? What are the 2 things stimulating the cells to clone? And what do they clone into? In the case of TA cells, they're going to become like cytotoxic T cells. And what do cytotoxic T cells do? So what's the stimulation to get them to clone? What do they clone into? And what do those clones do? What do they actually do to take out cells? Right? So you answer that question for cell-mediated, and you answer that same question for antibody-mediated. What's triggering it? What's getting it going? There's 2 stimuli. What are they? Number 2, what are they cloning into? So they're becoming plasma cells. They're cloning into plasma cells, and they're secreting antibodies. 3, what are the antibodies doing? So I might ask you that way, like, talk about antibody-mediated immunity, or I might just make a separate question, describe the functions of antibodies. So that's 5 or 6. I think I listed like 6 questions. So I'm inclined to make that your quiz only, at 1 chapter. I want a quiz, right? Yeah. So does anybody have any questions on this? I'm not quite done with the class. Sorry. But does anyone have any questions so far? Because we've only gone 40 minutes.
I don't, on the modulates, are you going to add some short videos for quiz? I think because we're starting quiz 3 now, material. Because I don't see any on their modulates.
Right, let me stop recording this first.